Hello, and welcome to this Fast Facts presentation from Cell and Gene Therapy Insights and Repligen. I'm Roisin McGuigan, an editor at BioInsights, and today we'll be learning about expediting downstream analytical testing. Our presenter today is Harold Elan. Harold has been with Repligen for almost 2 years, supporting biopharma analytical development, GMP implementation, and QC validation of slope spectroscopy methods and instruments for rapid bioprocess analytics. If you would like to ask Harold a question about his presentation, simply use the Ask a Question box to the right of the viewing pane. And with that, I'll hand over to Harold to introduce himself and kick off today's presentation.
Hello, my name is Harold Elan, and I'm a Senior Bioanalytics Application Specialist at Repligen in Germany. I am supporting our European customers during the implementation, method development, and GMP validation of our CTech analytical products. Today, I would like to give you an impression on how you can accelerate your analytical testing of viral vector bioprocess samples using our variable path length technology to expedite your downstream process development and eventually speed up your time to market. So thanks for watching this Fast Facts video, and let's go. So in this short video today, I'm going to give you an introduction about the gene therapy analytical solutions and the typical challenges and limitations. I will give you an introduction to our slope spectroscopy using the variable path length technology and give an overview about our CTech analytical systems. And in the end, I'm going to present to you a couple of gene therapy case studies to show you how you can use slope spectroscopy and benefit from it for your downstream analytical testing. Repligen is well known for transforming bioprocessing and addressing the critical steps in the biologic drug production. Our solutions offer high-impact technology innovation, flexible and scalable solutions, and high-quality, reliable, and robust products. We support our customers with regulatory documentation to support your agency filings and go into collaborative partnerships. You may be aware of our downstream analytical solutions and also the upstream products that we have. However, today, I'm going to focus on our CTech analytical solutions, which would be the CTech SoloVPE as an at-line system, and the CTech FlowVPE and FlowVPX as inline systems. So let's have a look first at how a downstream process of a classic viral vector looks like. So first, of course, you start with the upstream process, which would take about 7 to 8 days. And then you go through a series of downstream steps, starting with the harvest, lysis, clarification, the filtration, a couple of chromatography steps, and then the concentration, formulation, and other filtration steps, and the sterile filtration. So generally, these downstream steps typically just take a day. However, especially during the process development part of it, you need to test at each and every step for the genome and the capsid titer to assess also the full empty ratio, to understand how your process works and what you would like to improve in your next iteration of the process to get to your final process. So the goal here of analytical testing is to reduce the number of cycles, reduce the amount of raw material required, like, for example, the resins, which are very expensive, and ultimately accelerate your time to market. So what's needed for that? And that's, of course, you need to be able to make rapid development decisions using fast screening tests. And for this, you require in-process testing at every step in the process. So what are the challenges here? And the challenges mainly are, as I will show you on the next slide, is that most of these analytical methods that you can use to determine the genome and capsid titer and the empty-full ratio take much more than just a few minutes. So you can't do this as an at-line test. Normally, what you will have to do, or what many customers are actually doing, is they run through the process, take out those samples, and then analyze them afterwards, and then see what they can change and improve the next time to go through the process. But what if you would have a rapid test available that just takes a few minutes that you can do directly at line or even inline your bioprocess? So let's look at the current methods that are available for AAV quantitative analysis. So most frequently used is the combination of real-time polymerase chain reactions, so qPCR or digital droplet PCR, with an ELISA. So the qPCR is used to determine the viral genome titer, whereas the ELISA is used to determine the capsid titer. The caveat here is those methods come with an acceptable tolerance range of around 20 to even 40%. And the time to result takes at least a day for 2 analysts. Other methods that are seen as the gold standard is, for example, the analytical ultracentrifugation or the transmission electron microscopy, which are used to determine the empty-full ratio. But these methods can even take much more than a day, especially if they have to be outsourced. So basically, your time to results for such methods will be a day, at least up to weeks. So how can we address this using slope spectroscopy? And so first, I would like to give you a short introduction into how the slope spectroscopy method works. So slope spectroscopy is based on traditional uv-vis spectroscopy. In uv-vis spectroscopy, you take a single absorbance measurement, and the whole technology is based on the Beer-Lambert law, which tells you that the absorption of a specific compound equals the extinction coefficient of that compound multiplied with the path length, which is the way that the light travels through the sample, typically a 1 centimeter cuvette, and then multiplied with the concentration. So the path length here is a fixed value, and the concentration is the variable that needs sample dilution in case you have a highly concentrated sample, because otherwise your sample or your measurement will be outside of the linear range of the spectrophotometer and of the Lambert-Beer law. So how do we address this with variable path length technology? So in variable path length technology, we take multiple absorbance measurements for slope-based concentration results. So instead of shining the light through the side of a cuvette at a fixed path length, we shine the light through a fibrette, which is a piece of fiber optic that goes directly into the sample. The sample is in a sample vessel, which is sitting on the detector. And by being able to vary the path length and moving this fibrette up and down, we can actually vary the path length and take multiple measurements at different path lengths. So here, the path length is a variable, capturing multiple absorbance measurements in less than a minute, and the concentration can stay fixed and does not require any sample dilution or adjustment. So how does it actually work? During a measurement, the system will first go to the 0 path length at the bottom of the sample vessel, and then an algorithm will find the path length where you would expect 1 absorbance unit. And then it will plot this absorbance over the path length and take the first data point. Subsequently, it's going to collect up to 10 additional data points at shorter path length. And then you can create a linear regression through the series of absorbance over path length data points, and that will give you the slope of that linear regression. The whole data acquisition takes less than a minute, but ultimately the slope m will equal the extinction coefficient multiplied with the concentration of the sample. So by measuring the slope, you can easily assess the concentration. And that means we can go to very short path length for very high concentrated samples, or very long path length for low concentrated samples. And the additional benefit, even if your sample is low concentrated, like the viral vector, you will still get an R² value to tell you what is the quality of your data, because the R² value will give you a readout for the accuracy of the linear regression. And with that, we have a built-in data quality and robustness testing. But even for high concentrated samples or high absorbing molecules like nucleic acids, you can use slope spectroscopy for a much broader concentration range. So if we talk about monoclonal antibodies, and this is where the SoloVPE has become basically the gold standard for at-line testing, but also release testing, we're talking about concentration ranges from 10 microgram per mil up to 300 or even higher milligram per mil that we can measure without any need for sample preparation. And that makes it a very easy tool to implement as an at-line test on the manufacturing floor, because it's so easy to use and the manufacturing scientists can just use it and don't have to send the sample out to analysis. So looking at the comparison between traditional UV and the variable path length slope spectroscopy, we are reducing a traditional 7-step process, which starts with the estimation of your sample concentration, the error-prone dilution step, and then you measure, plot, calculate, accept the criteria, and report the results. And you can just replace this with the measurement and the reporting, which is done automatically through our VPT software. The two systems that we have available are the SoloVPE as an at-line system, where you take the sample, put it into a sample vessel, and measure it at line. But we also have a FlowVPX system, or the older FlowVPE system, which have a flow cell that you can directly attach to your process, for example, at a UFDF step or a chromatography step, to measure your sample in real time. So by implementing the VPT technology, you will remove or automate multiple steps. You gain time to results, robustness, and simplicity. And this encapsulation improves your workflow and validation. So let's have a look at how this technology applies for gene therapy. And I will show you a couple of case studies for gene therapy. So as I mentioned before, slope spectroscopy is already very well established in the map world, measuring protein concentrations. However, these are not the only applications where you can use the SoloVPE for. There's a number of additional applications, including applications for gene therapy. So I'm not going to go into details on all of these, but just to give you an overview, you can use the SoloVPE for culture density with the classic OD600 method. You can use it for plasmid concentration and plasmid purity. You can use it for other nucleic acid modalities like mRNA vaccines, mRNA therapeutics, oligonucleotides, like antisense oligonucleotides, or any other nucleic acid-based modality to determine the concentration. But by, for example, calculating the A260 over A280 ratio, you can determine the purity with regard to protein contamination, or even looking at A260 over A230 to look for purity regarding salts and solvents. You can use it to determine viral titer or empty full capsids, and that's what I'm going to focus on in the remaining part of this presentation today. But you can also put it at line or inline for UFDF monitoring. You can use it for PANOVA filter integrity testing. If you combine the FlowVPE or FlowVPX with the MALT system, you can use it for real-time molecular weight and also for drug product formulation. So as I mentioned, in the following part of this video, I'm going to focus on the empty full capsids and viral titer applications for the SoloVPE and FlowVPX. However, if you are interested in any of the other applications, please go to our website or reach out to us, and we're happy to give you more information. But there are plenty of publications on all the other applications on our website. Just have a look. So in the remaining part of the presentation, I'd like to introduce to you the applications for viral vector production. So let's have a look at the very first feasibility study that we conducted with a customer to see if we can use the SoloVPE to measure the titers and empty full ratio of a sample that was a full sample of AAV or an empty sample of AAV. So what you see here are actually the regression lines of the slope absorbance measurements for the A260 and the A280 measurements. So you can see we can get excellent linear data, measuring a slope value that we can use and getting an R-squared value of 0.9999. So this is excellent R-squared values showing us that we have high quality data. And we can also see that there's a difference between the empty and the full samples. So using those slope values in calculations that are accounting for the spectral overlap between the A260 and the A280 protein and viral genome absorbance values, we end up with the viral titers. And then just looking for the capsid titer and the genome titer, you can calculate the percent full ratio. Using the SoloVPE system here, we ended up with a percent full of 78%, whereas the qPCR ELISA method ended up at 69%. So this is pretty close. However, the qPCR ELISA method came along with a tolerance of around 20 to 50%. And the results took about a week to be generated, sending out this to a specific analytics lab. However, in the SoloVPE, the tolerance of the slopes was just plus minus 2%, and the time to results just took 5 minutes for a triplicate measurement. So this shows that the SoloVPE can be used to determine empty full ratios for AAV samples. After that, we actually went into a number of additional case studies, and I'm just focusing on one here for the sake of time. And this is a study that we did together with Vigene, who is generating and manufacturing and providing AAV reference material. So we tested AAV8 and AAV9 samples that were regarded to be empty or full capsids. And using the SoloVPE with the method that I showed on the previous slide, we were able to determine the viral genome titers and capsid titers and calculate the full and empty ratio. And then comparing this with the full empty ratio from qPCR and ELISA, you see that we're actually pretty close in most of the cases. So this showed that there's an equivalency of both method results compared to current PCR ELISA method, well within a plus minus 40% range or even lower. The DNA and capsid concentration was measured in a single test in minutes versus days. And we also found that these Vigene certified reference standards can be used as a universal system suitability control. Then we went into another case study with a customer to see if we can use actually our inline technology to monitor a UFDF process. So here, our SoloVPE was actually put at a UFDF step to measure the concentration and the viral titer of the VG per ml, actually, to the viral genomes during the UFDF step. So if you just have a look at those data first, you see that the data match pretty nicely to the digital droplet PCR results the customer obtained after the UFDF step. So this data was generated in real time. And you see, especially during the diafiltration and in the final drug substance stage, the difference between the two methods is really low. But more than that, you can even use it to really monitor the UFDF step in real time. And we see just after the concentration step or towards the end of the concentration step, the scientists actually found some funny spikes here in the profile. So they had a closer look and found that there was some cloudy solution and that their product was probably starting to aggregate. So they're slowing the mixer to get back into a kind of steady state. And then they could start the diafiltration process. So using the SoloVPE here to monitor a UFDF step for an AAV process, this found that they could know the AAV titer without a complicated and time-consuming ddPCR and know this titer in real time. And then also it helped them to carefully concentrate up the product without damaging the precious product. So how does this technology actually fit in now on a typical AAV downstream bioprocess? Showing just the slide from above again, to see where it actually fits in, we would say it's most suitable actually for the later downstream part in the process. So the reason for that is that here in these parts, you are having highly purified material. Early in the process, you will still have a lot of host cell impurities like host cell DNA, host cell protein. And since uv-vis is always an unspecific method, you would also measure those impurities. So that's why we think it fits in much better at the later stage of the process when you have at least some purified material. And also, we found in our case studies that we typically have a lower limit of quantitation in the range of the 1E11 viral genomes per milliliter. And the concentration at these earlier steps may be lower. So, but usually during the later downstream steps, this concentration is actually high enough to measure it with a SoloVPE, and also the purity is sufficient. So that means that you can use the SoloVPE and the FlowVPX systems as a rapid and highly reproducible in-process screening method for AAV titer and empty-full ratio to reduce your number of cycles and the raw material required. It is a simple, quick method for measuring process recovery during early process development. And you can transfer it very easily with high level of system-to-system consistency. And of course, all our systems come along with the 21 CFR Part 11 software features to enable to use the system in GMP environments. So we are committed to your success. And we, of course, will help you to develop the methods, to implement those methods, to validate them through our team of global bioanalytics application specialists. So the whole platform can be aligned across your different process steps. So from cell culture to harvest to purification, formulation, and fill and finish, you can use either the SoloVPE as an at-line concentration measurement or the FlowVPX as an inline concentration measurement. And it's a single platform also across all your different functions, like QC, analytical development, MSAT, manufacturing, and the process development. So thank you for listening to this Fast Facts video. I'm Harald Ehlen, and I'm happy to answer your questions if you just reach out by email. Or I would like to recommend to you to go to our website, ctech.repligen.com, to have a look for further information, and please reach out to us. Thank you.
Thank you, Harold, for a great presentation. And thank you to our audience for watching along today. If you'd like to download a poster version of today's talk, summarising the main points, you can find this below the video. We hope you'll join us again soon for more Fast Facts.